Isa. It's my great pleasure to introduce Henrik, whom I know from quite far back because Henrik did his master thesis at the University of Copenhagen in the group of Signe Larsen, where I was also in fact working at that time. So I know Henrik from then. After his master thesis, he moved on to Switzerland, where he worked with Professor Dieter Schwarzenbach, and during this time he became a user of synchrotrons and large scattering facilities, and I guess a hallmark of his research from then on. He has, after that, done a postdoc position also at Lausanne in Switzerland, and he has been in the US at the University of California, Santa Barbara. And after that, he came to Aarhus, where he has been since then. He has started out as a research assistant and is now a full professor at the Department of Chemistry at University of Aarhus. I think his research has moved from in the very beginning to be a combination of charge density studies with theoretical calculations, moving through more material science applications. And I think it's fair to say also that during the later year, he has also been moving into more biological materials, in particular in studies of bones, and also perhaps not just doing hardcore diffraction, but also using imaging and related techniques a lot more. So I hope you agree on all this, Henrik, and I will pass the word on to you now.
Well, thank you very much for the kind invitation and for the very kind introduction, Mikael. I hope you can see my slides now. Yeah, I still remember as a student, one of the things that really turned me on to X-ray techniques was in fact a lecture given by Mikael, where he was talking about the Patterson function. And this still stands out very vividly in my mind as something really defining in my career. So thank you for that, Mikael. And thank you very much for the invitation and for coming to hear about our work today. As Mikael say, I have been sort of on a journey from over material science and now perhaps transgressing more into trying to also contribute a little bit to some biological questions. And I hope I can convince you that we've been at least able to do some contributions in this area. So today I will mostly, or I will be exclusively be talking about how we can study bone structure. But many of the techniques that I will present are actually also potentially useful in many other biological scenarios. It's perhaps something we could talk about during the Q&A session. But first, I would like to thank the people involved in the work. So these are predominantly people from my wonderful group. And today I will mostly be presenting work by Nina Wittig, Maja Østergaard, Jonas Palle, Fiona Bach-Gansmo, Mi Birkbach, and Simon Fröhlich. But everybody have pitched in along the way. And some very good collaborators in the work today at Aarhus University and also at McGill. And then very importantly, at the large-scale facilities without whom simply would not be possible to do this work. Okay, so as Mikael mentioned, I'm a materials chemist of origin. And you might wonder, why is a materials chemist studying bone? And we're interested in bone for several reasons. One is that there are some very important unanswered biological questions that we are interested in, but also because bone is a very fascinating material in itself. So if we think about the way nature constructs materials, it's really quite different from the way we in our technology construct materials, in that nature uses hierarchies. And so bone is an excellent example of this. It is formed by collagen fibrils that are synthesized intracellularly and then induced to aggregate into fibrils extracellularly. Once the fibrils are formed, they become decorated by apatite nanocrystals to form a composite material. And it's only through the proper arrangement of crystals and fibrils that bone gets its important biological functions, which is predominantly to provide mechanical support. However, this hierarchical structure does not stop here. There are then additional motifs. And if you focus on the right-hand side of this, you can see a slice through what could be a sketch of a human bone, a human long bone like your femur. We and a few other animals are a little bit peculiar in that we have a repair system to the bone that's called haversian remodeling, where osteoclasts, they drill long tunnels into the bone where the bone-forming cell osteoblasts, they reform bone matrix from the outside in after on. The reasons why we have this haversian remodeling and other animals like rodents do not is really a major outstanding question in the field. And basically anything below the 10 micron scale is something people really discuss quite intensely. So for instance, what is the size and shape of the crystals? How are they arranged in relation to the collagen fibrils? And all this is stuff that we are still working on trying to understand better. Bone is rather interesting in the sense that its biology has changed quite significantly over the past few decades. It used to be considered maybe a dead material that you would just kind of walk around on. But it's been realized that the cells inside bone, osteocytes, some of the longest living cells in our body, they actually communicate with other organs. So the view has changed that bone is this sort of dead graveyard in biology, that it's actually a very important endocrine organ that has important crosstalk to other organs, for instance, in obesity, diabetes, etc. So we've been interested in trying to understand the cellular network within the bone, not so much the cells themselves, but rather at a more modest perspective in the beginning because of the technical challenges involved, just how the network, porosity network inside the bone matrix occurs. And for this, we will use X-ray imaging. Then to address the mineral crystals in bone, we can use X-ray diffraction and X-ray fluorescence, and I will demonstrate to you how we can do this in 3 dimensions so that we can really pinpoint where exactly within the bone matrix we measure, we obtain information about the crystals. And you will see that these are not, as one might thought, one size fits all, but they're really important variations as you move across various structural motifs in bone. So what we would really dream of doing is to have a single experiment where we could just zoom in basically to the cellular scale in a piece of bone that would be very large. Unfortunately, that's not really technically possible yet, but we're getting closer. So in the laboratory, you can do 3D imaging like you know from a CT scanner in a hospital, and then we can get images maybe with resolutions down to, say, a handful of microns or so. So that's good enough to look at mechanical turnover in bone, but it doesn't really tell us anything about the cells. So to look at the cellular cavities, we could use X-ray microscopy in the laboratory, which is a rather laborious and slow process. Or we could, as I will show in the coming slides, use synchrotrons, where we can do high throughput measurements like you can see here in the center, where each of these green blobs houses a single cell, and the red correspond to a blood vessel. And it's even possible to obtain nanoscale resolution in 3D, where we will see that we can use this to unravel new features of the cellular communication network. So the workhorse technique that we are using in my laboratory now is tomography. And the idea is really rather simple. It's not enough to look at a problem from one side, but if you look at it from several different sides, you can, a bit like in Sudoku, where you make a puzzle of various entries, you can back calculate what should be the values inside each little box. We can do exactly the same based on images. So we take an image, we rotate the sample, take another image, and by this we build up a data set as a function of position across the sample and viewing angle. And from this you can back calculate what will be the structure inside the material. So we've been doing this through a number of years. And I will start with sort of modest length scale, where we use pixel sizes in our images. They're called voxels in 3D, but basically this corresponds to the pixel size in your cell phone camera of roughly, say, 300 to 400 nanometers, and that gives a sub-micron resolution. Now, doing this, we can measure many samples, which is extremely important when we're trying to do sort of organ scale biology, because there's a lot of biological variation. So we need to be able to do statistics. And say, with the use of, say, a graduate student sample changer, you can go through a few hundred specimens in a couple of days, and so actually collect statistically relevant data sets. So we've used this to analyze the osteocyte housings, so-called lacunae, the size and shape in various different types of bones. And I'll just give one example here that you can see on the right. So this is a cross-section through a rodent bone. This is from a rat. And you can see in blue, these are cavities corresponding to the blood vessels. Then there are the light orange and the dark orange discs, and these are the osteocyte lacunae. And so rat bone is different from human bone in that they do not have this Haversian remodeling process that I mentioned. So that means that there's the sort of remnants of the originally formed bone, and that's the central part that you can see with the light orange osteocyte lacunae. They are formed by a process called endochondral ossification, by which the long bones are formed. And this is a more sort of disordered way of making the material, where you first form a cartilage template, which is then turned into bone. And you can see that the osteocyte lacunae are organized in sort of mumble-jumble fashion in this region. Outside, the bone formation takes place by apposition of collagen fibrils that are predominantly oriented with the long axis of the fibril along the long bone. So that means that the bone becomes lamellar, and we can see this reflected in the orientation of the osteocyte lacunae. So what we could show in this work was that the orientation of the osteocyte lacunae, but also their shape, was significantly different from the central endochondrally derived bone type in the center from that further away, reflecting the fact that these two types of bone are in fact quite different. And also that one should be quite careful using rodent long bones as models of human bone. I'll just give one example of a biological question that we have been trying to address. And one of this is the role these osteocytes may play in biology. So it has been proposed that they should be able perhaps to lyse the surrounding bone to contribute to calcium homeostasis in the body. And that has been debated since it was proposed in the late 60s, and quite vehemently so, in fact. It's a rather interesting literature to look at. And it's been proposed that if you challenge the body so that you would need to mobilize calcium, that you should see enlarged lacunae as a response to this calcium mobilization. So we've been studying it previously in rats, where we Botox immobilized hind legs, but we did not see any effect. If you are then a mouse and you have a litter of young, you really need to mobilize lots of calcium for the milk. And so it has been reported in CD1 mice and C57BL6 mice that enlarged lacunae occur upon lactation. So we wanted to study this process in more detail in 3D and used another mouse strain called NMDI mice. And so we conducted tomography, and you can see that we nicely see the osteocyte lacunae in yellow. But when we measure the size of the lacunae that you can see in the top right panel, these are the lacuna sizes. In this histogram, you can see that's a certain variation, but we see no difference between the lactating group and the control group. An advantage of using the synchrotron is that we can actually quantitatively calculate the mineral density in the surrounding bone, and we can also see that this is not different either. We did complementary measurements using electron microscopy and light microscopy, and again saw no difference. So this indicates that NMDI mice do not display osteocytic osteolysis, which makes us wonder how general this biological property that has been proposed of osteocyte is. We simply do not know yet. So this is nice, but it doesn't really allow us to provide information of how cells communicate within this cellular network. So for that, we need higher resolution. And there are a number of techniques that you can use for this, but they both make use of an important feature of synchrotron and free electron laser X-rays, namely the fact that they are coherent. And when you have coherent, then refraction becomes a very important part of what happens. So that means that we can build images where we make use of the refraction effect to build the image. This is very nice because the refraction effect is roughly 2 orders of magnitude larger than the absorption effect. So we can measure much finer details if we can just harness this. And so there are 2 sort of main ways of doing this. One is called tachography, which I will not talk about. And another one is called phase propagation holotomography, or say phase contrast tomography. So in phase propagation holotomography, you focus the X-ray beam, which provides an optical magnification, and then you place the sample at various distances between to the detector. And when it's very close, you measure mostly absorption. When it's very far, you measure the diffraction pattern. And so the resolution you potentially can obtain is defined by the focal point. And at the leading facility for this, which is ID 16A at the European Synchrotron in Grenoble, they're really very good at focusing hard X-rays. So you can get down to maybe sub 20 nanometer focal points and hence potential resolution in this way. So these are results from mice bone. And it's basically the same data set just shown in 2 different ways. So I've carved out a couple of cellular cavities because otherwise you basically wouldn't be able to see any details. So if we focus on the left, you can see these 2 cellular cavities labeled 1 and 2, and see how they're interconnected by these plethora of tubes. And these tubes are only a few hundred nanometers in diameter and termed canaliculi. So it's within these canaliculi that the cells are believed to extend their, or are known to extend their cellular processes for cellular communication. When we were looking at these data, we got very surprised by the fact that there are many bulges on these. And so we were very intrigued by this because this has not been reported by before, and it's basically impossible to see in 2D data. So we looked more carefully at these, and we saw that they occur in places where several canaliculi cross, as you can see in the image on the left. So this would suggest that there are sort of crossings in this canalicular network. So we label these features junctions, canalicular junctions. And for this to be functionally important, it would have to be in contact with the fluid flow possibilities within the cellular network. So it's believed that these cells, they sense mechanical stress by changes in the shear fluid forces, flow forces within the canaliculi. So of course it would be important if there was sort of a little bloboid in this network. So to test if this is really biologically in contact with the fluid dynamics of the bone, we use confocal laser scanning microscopy. So we placed pieces of bone in the solution with dyes, and the dyes is then infusing into the void space of the bone, allowing us to see which part is connected to the network. And so we could see that the junctions occurring in mouse, rat, cows, and humans were all connected to the cellular network, meaning that they are, in fact, part of the fluid flow network of the bone. And we see quite a lot of these. So in mice, it's roughly 1 junction per osteocyte lacuna. So there are really quite many of these. So what could they possibly be doing? Well, one intriguing possibility would be that they act sort of like in a highway network as a junction for multicellular communication. Of course, this is pure speculation at this point. We would have to actually have biological proof for this, but it's a very tantalizing possibility. What is for sure, and we in this publication cited below, we provide modeling evidence for this, is that the fluid flow within the cellular network is heavily modulated by the presence of these bathtubs within this network. So a bit like if you were to have a lake in a network of rivers. So that's important because it would change the stress sensing capabilities of the cells and the forces that are possible to sense. The biological impact is something that we're trying to work on presently. So this is nice, and we learn a lot about the void spaces within the bone. But what about the surrounding bone matrix? Can we also learn something about this? So to do this, I'll go back to illustrate this. I'll go back to the osteons. And so now we go back to human bone. So the osteons, they are built outside in. So that would mean that if we could look at the signature of the apatite mineral within the bone, we could sort of sample the biomineralization process history by spatially resolving the osteon structure. So this is fairly challenging because you would need to collect diffraction patterns from these nanocrystalline apatite crystals within the bone matrix with a resolution that's better than the structural variation within the osteon. So that means subcellular resolution. So we would need resolutions better than 0.5 micron or so. And so this is actually not so easy as it sounds, but we gave it a shot anyway, and we're actually quite surprised by the results. So the experiment we do now is a little bit more complicated. We focus the X-ray beam into a pencil beam, and then we build a 2D image by raster scanning the specimen through the beam. So now we have a 2D image of the specimen. Then we rotate it, measure again, rotate, measure again, and then we build up a tomographic data set just like for the full field imaging I described before. Then we can mathematically reconstruct what would be the diffraction pattern from inside the sample. And you can do the exact same with X-ray fluorescence, which provides you the signatures and thereby concentrations of heavy elements, such as in our case, calcium and strontium. So to do the experiment, we used 400 nanometer wide X-ray beams. And we made for technical reasons, two specimens, one for the fluorescence experiment, which is a cylinder perpendicular to the osteon canal, and one for the diffraction experiment, there was a disc parallel to the osteon canal. So let's look first at the fluorescence results. So what I've shown here in green is the calcium concentration, and in yellow, the strontium concentration. And they're shown in two slices through the reconstructed 3D volume. And to the right, you can see the void spaces. So the osteon canal is at the top of the image. And if we focus first on the calcium, you can see that we see osteocyte lacunae as these black voids. And there's maybe even an indication that we can see the beginning of the canalicular network, indicating that we for sure have submicron resolution in the images. At the very top, you can see a higher calcium content close to the osteon canal. And then there's a more or less constant calcium concentration onto a transition zone roughly 3/4 of the way down in the image, where we transition from the osteon to another type of bone called interstitial bone, which is known to have a higher calcium content. In the same experiment, we also measure the amount of strontium. And strontium can substitute into the apatite lattice and is an important anabolic agent for bone formation. So what we can see is that strontium is actually, in this particular case, placed outside the osteon, so predominantly in the interstitial bone, but also with varying concentration across the osteon. So I should say that this is a slightly longer experiment. So this is a one-off. We just have the one individual that we studied here. So of course we need to be careful not to make too broad statements about the biological significance of this finding. So the key take-home message here is that strontium is very heterogeneously distributed within bone. And also we can see that close to the osteon canal, the calcium content shown here in this distance plot is quite significantly increased, meaning that the amount of mineral close to the osteon canal is higher than further away. Then let's look at the diffraction. So there we collected a small volume with a height corresponding roughly to a bone-forming osteoblast in size. So we measured 3.6 million individual diffraction patterns, And each of these we refined a model against using a technique called read-weld refinement, and a little program we have written that helps us do this automatically, so we don't have to sit and fit each one by hand. So in this way, and with a moderately good computer, you can do these in a few days. Today, when we did it originally, it took us 3 months. So what do we learn from this? Well, we can get the amount of crystalline material which is present, and a lot of other stuff we can get out. I will just focus on one feature, namely the peak width, which tells us something about the crystallite size and or possibly something called microstring fluctuations. But the dominant factor here is the crystallite size. So if we look first at the amount of crystals, I show you here the disk, and so the osteon canal is at the top. And what we can see is that close to the osteon canal, there's an increased density of crystals, just as we saw an increased calcium content, so the two go together. Secondly, we can see pseudo-periodic variation in the mineral content, which reflect the fact that the mineral content within the osteon varies spatially. So this is very important for the mechanical properties of osteons, because it leads to the fact that there's a mechanical varying stiffness, locally varying stiffness within an osteon, which is very important for the toughness of your bones. If we then look at this microstructural characteristic, then it becomes a little bit complicated because bone crystals are anisotropic in shape. So we see that we can get information about this from the data. But suffice to say is that we can see that there's a spatially varying crystallite size across this osteon. So the crystals are not the same across. And most importantly, close to the osteon canal, we can see that the peak widths are broader than further away, which reflects the fact that the crystallites are smaller. So close to the osteon canal, the bone is different with smaller crystals and a higher crystal concentration. So how can this be? Why are the mineral crystals smaller close to the osteon canal? Well, one theory could be that this has to do with the biomineralization mechanisms. So when bone is formed, there's a set of proteins that help control the mineralization process. And one of this is osteopontin. And osteopontin is an important inhibitor of mineralization. And it's overexpressed around blood vessels, the osteon canals, and osteocytes. And so what happens in normal bone formation is that there is an enzyme controlled by the gene FEX that breaks down the osteopontin and thereby allows the mineralization process to proceed. If you do not have FEX, then you cannot do this osteopontin breakdown, and you get a disease called cross-linked hypophosphatemia, which is not fun, I can guarantee it. In humans, this is fortunately a rare disease, but if you are a mouse of the strain HUP, then you're born with this disease. And so you can see what happens on the right here. These are tomographic results obtained in collaboration with Mark McKee from McGill. So we have the normal wild-type mouse on the left and the HUP mouse on the right. And so the light gray are places with lots of mineral, normal bone, and we can see the large dots and small dots. So the small dots would be the osteocyte lacunae, and the large dots would be blood vessels and canals. So in the HUP mouse, you can see that these canals, and at higher resolution, you would also see around the osteocyte lacunae, are heavily enlarged and surrounded by unmineralized matrix. So this suggests that when you cannot break down osteopontin, the region around the osteon canal is not mineralized, reflecting that this is really a different type of bone which is present here, heavily influenced by osteopontin-like proteins. This also happened around the osteocytes. So here I show in color, these are the osteocyte lacunae in wild-type mice and in HUP mice. And you can see in the color scale, that's the size of the lacunae. So we can see that they're really very much enlarged in the HUP mice. So to quantify this, we did an analysis that allowed us to extract the osteocyte lacuna itself and the surrounding region around it that we term the unmineralized pericellular matrix. So what we can see is, if we compare the wild type mouse to the Hüb mouse, is that not only is the lacuna volume itself increased from roughly 225 to 325 cube microns, but there's also this halo around the cells, which is rather large in these Hüb mice, and that reflects exactly the human condition, in fact, in this cross-linked hypophosphatemia. So what we learn here is that during the bone biomineralization mechanism, the proper processing of mineralizing controlling proteins is extremely important and can result, if failure is here, it will result in rather severe diseases like this cross-linked hypophosphatemia. Okay, so that's very nice, but as you probably know, bone is very anisotropic in its mechanical properties. So if you, when you stand up straight, you don't normally fracture your bones, but if you fall on the side, especially if you have frail bones, then you have a very enlarged risk of fracture. And that's because bone is designed to carry the compressive load of our body weight. And this anisotropy in mechanics comes from the fact that the collagen fibrils are organized in space. So to fully understand the properties, we need to understand how mineral and fibers, collagen fibrils, are oriented in 3D. So how can we do this? This turns out to be rather difficult, but it turns out that you can use the fact that small angle X-ray scattering is anisotropic. So this is the signal very close to the beam stop on the image on the right. I will just quickly point it out. So this would be the small angle scattering signal right here. And you can see it's not circular in shape. This curve reflects the orientation of the collagen fibrils more or less. Formally, it's the orientation of the nanoscale structures within bone, but it translates more or less to the collagen fibrils. And so if we now do this tomographic reconstruction and then view the sample from several tilt angles, so we tilt the specimen, rotate, and raster scan it, then you can build up a SACS tensor tomography data set, which was proposed by Mariana Libby and other people about 5 years ago. So that's nice. That tells us something about the collagen fibrils. But what about the mineral itself? Well, to do this, we extended this method to also include the wide-angle X-ray scattering signal, which are these rings you can see out here. And you can see that this one in particular, the sharpest of them, which is the 002 reflection of the apposite lattice, is also oriented. So we looked at now at data from a human. So this is from the iliac crest, where we extracted a part of the iliac crest containing lamellar bone. And in the bottom, you can see the sample that we were working on. So there you can see the collagen fibrils, mineralized collagen fibrils themselves. You can see here that there's sort of a co-orientation this way and a different orientation in between. So these are individual bone lamellae we could trace along the specimen. When we then looked at the projection data, we could see that mostly this 002 reflection in the red circle was perpendicular to the SACS signature. And that's exactly what you expect from the models we have of bone. But in some cases, like the one shown here, there was a misorientation between the two. And so to do this experiment, we used a 1 micron X-ray beam. So the question is this now a projection feature, or is this really true biology? And so to allow doing this, we did this tensor tomographic reconstruction of the two features. And so the results come here. So here's just again the scenario. We're looking at this laminar bone cube. And then we reconstructed the orientation of the collagen fibrils in the top. And you can see these as these little bars. And the darker the sort of the orientation of them shows you how they are oriented. And then we did the same for the crystals within the bone. That's shown in the center plot. And these should be perpendicular to each other, aligned in just the right way for the current bone models to really match. So you should expect, if we calculate an alignment feature, which we do in the bottom, that all the voxels in the image should be red. But we see that quite systematically, let me just get a pointer here, there are places within the bone, here and here, for instance, where there's a misalignment between the two scattering features. And so this could be due to various different features. One being some rather technical features about how the scattering experiment is done. What we favor as a more likely explanation is that locally is a small contribution beyond these mineralized collagen fibrils that I explained in the beginning. And so what we then observed is these misalignment regions, they occur in regions that correspond to what has been labeled disorder collagen motifs. So when we look at the stacking of collagen fibrils in electron microscopy, we can see that they are mostly very ordered, but sometimes there are disordered regions in between. And within these disordered regions that the osteocyte lacuna canalicular network runs with the canaliculi highly concentrated in these disordered motifs. So what we are working on right now is a hypothesis that perhaps this misaligned bone fraction results from the presence of canaliculus associated mineral, which might not be tightly associated with the collagen fibrils and hence have a different orientation distribution than the collagen fibrils. So we're trying now to see if we can prove this hypothesis. Okay, so I will begin to wrap up now. So what I hope I have convinced you is that with multimodal tomography, we can get lots of insights into biological samples. I have focused in this talk on bone, but I'm happy to discuss possibilities to look also at soft tissues. So with the synchrotron, it's possible to do submicron tomography on hundreds of samples, which allows you to do proper statistical investigations. We can obtain nanoscale resolution, allowing us minute details of, for instance, multicellular networks. And we can use other contrast modalities. And with this, I showed that there was canalicular junctions in the osteocyte lacuna canalicular network, how mineral crystallography depends on positions within the osteon in human bone, reflecting the mineralization conditions, and finally, how we can extract orientation information in 3D. With this, I would like to thank you very much for your kind attention, and I'm very happy to take any questions. Thank you.
Thank you, Henrik, for an excellent talk, I think, demonstrating how far you can go by combining many different scattering techniques. I think it's really beautiful work. If you others present here agree with me on this, I'll mention that we